All right, so let's begin looking at physiology and what exactly is physiology. It's basically the study of the body's function. How does this relate to anatomy and physiology? Well, anatomy is the structure, and physiology is more the function of that anatomy. Okay, but they go kind of hand in hand because anatomy is set up to serve a certain function, to serve the physiology of the body, of how it's functioning. So when we're looking at this normal functioning, right, and so essentially looking at the physiology, we have to take in chemical components and physiological processes. Okay, you'll see on anatomy, you guys going through anatomy, you spend a lot of time at tissue level and then up to organ levels. On physiology, we're going to be down at the atomic level and the cellular level, looking more at that function. And the way these cells operate and how the body operates usually is through chemistry. So I know chemistry is not a requirement, but we are going to definitely have to be using some chemistry. So our next lecture will be basically on that chemistry we'll be using throughout the semester. And another point between anatomy and physiology is that anatomy you can kind of memorize everything and then put it aside or drop it and then memorize the next. In physiology, the stuff I'm going to be teaching you here at the beginning, you're going to be using throughout the semester. So this beginning stuff with chemistry, with cellular respiration and enzymes and stuff like that, these are all, in membrane transport, these are all items that you've got to get down because throughout the semester you're going to be seeing the same concepts because basically these components are how, or processes are how the body works. Okay, so I need to stress that is that you need to get these beginning components down and get the concepts down because basically the whole body uses these concepts to basically perform the normal functioning of the physiology of the body. And as I already said, our physiology is rooted in chemistry and physics. Lots of chemistry because basically chemical reactions is how the body's making everything take place by making energy, making the neurons work. This is all chemistry and chemical reactions that are making this take place and making basically the survival of your body, right? So physiology, we are definitely going to be taking in chemistry. I don't mean to scare you, but I do mean to scare you that I really am you've got to get the chemistry down or the concepts down. Again, it's kind of baby chemistry. I'm going to give you the components that we just need. We're not going to be going through a whole chemistry lesson, but I'm going to give you the nuts and bolts of chemistry that you're going to need to use in this physiology class. Physics, also because you think the heart's like a pump, we're using physical components and kinetic energy and stuff like that. We will get into basic components of that. So there is some physics involved, but the big component is going to be chemistry because everything revolves around chemical reactions in the body for the most part. Through this semester, we're focusing on physiology, basically the normal functioning, but since most of you will be going into the medical field, we will also focus on some pathophysiology. Pathophysiology is basically where disease affects the physiological process. So we'll focus on some certain diseases to help pinpoint the normal functioning. Because both of you and most of you that are going in the medical field, basically you're going to be treating pathophysiology, unless you're in internal medicine where they're just coming in for a physical, but most patients are coming in with some kind of disease state or problem where the pathophysiology would be involved. Just showing one common state, pathophysiology, Alzheimer's disease. Okay, you got your neurons here. Hopefully this looks familiar from your anatomy class, but typically you don't have these large plaques here, but in Alzheimer's, this is one of the key characteristics or components of it. So let's get into the heart of physiology here. and what we're basically going to be focusing on through most of the semester, and that is a word called homeostasis. Okay, homeostasis. Basically, as an individual, you keep your internal environment stable. Okay? When you go in to the beach, say it's 60-degree water. Right? When you go in there, your temperature of your body doesn't go down to 60 degrees. It doesn't match the outside. You constantly keep this internal temperature. So you're keeping that internal temperature homeostatic, or going through homeostasis. Okay, so this is the main function because this is what keeps the body functioning. We have to keep certain variables in homeostatic range for those chemical reactions to run all the body to be able to perform the processes that have to take place. They need this kind of constant internal environment that's within these certain ranges. Okay? Probably one you're common with is blood sugar. Blood sugar we keep within a certain range. That common disease that we know about, diabetes, that is a disorder in which the blood sugar cannot be brought down or back within a homeostatic range. Okay? The thing is, it's maintaining a similar condition. This range, though, is not like here a flat line. You can see it's going up and down, up and down, up and down, but it's not taking these big broad strokes. We're not going way up, way down. We're keeping within a certain range. It's not a flat line, so homeostasis isn't, okay, my blood sugar's at 100, 100, 100, 100, 100. It goes from 80, maybe up to 110 after you eat a small meal, or even up to 140 if you eat a bag of Skittles. Okay, but then it brings back down, around 80, 90, okay, say around 90, and it keeps fluctuating within this range, but it's not going up to 300 and then back down to 50. It's staying within this nice homeostatic range. So it's stable. It's not so constant where it's just one number, but it's within this stable range. And that is what homeostasis is. This is what a lot of your energy goes towards, is keeping this internal environment homeostatic. So the body, your cells can all run properly. Your cells of your body need a proper working environment to be able to perform the function that they need to perform. So since we have to keep this internal environment homeostatic, let's look what actually makes up this internal environment. Okay, we can have the external environment, what's outside. We got air outside, cold air. If you think of your digestive tract is actually an outside environment. Until that food actually crosses over a layer of cells, it's not into the internal environment. So this is all external environment. Okay, and to keep our internal environment homeostatic, we got to exchange things. We got to get things in, right? We need to eat sugar, so we have to keep our sugar levels high because our body's constantly using sugar. So we have to exchange. We got to get sugar in. But stuff like waste material, stuff like uric acid or urea, we have to remove to keep, again, a constant homeostatic environment. All right, we want to keep our urea levels at a certain homeostatic level in the internal environment. Okay, so we have these cells. We have exchange cells, protective cells. So these cells, that line are facing the outside environment. Those would be your epithelial cells. To get something in or out of the cell, you have to cross over a layer of epithelial cells. Okay, exchange cells, we can think of our digestive tract. We have epithelial cells that line our digestive tract, and they do absorption. They absorb nutrients and bring it in to what we call the extracellular fluid. Okay, the extracellular fluid, that can be blood. Okay, it's anything outside of the cell, extracellular, outside. Okay, And we have, basically this is made up of two components. We have blood, but there is also fluid that surrounds the cell. Okay, so we have blood and we have extracellular fluid, ECF, or you also hear ECF as interstitial fluid. Okay, but your body, your cells, these are the functional components of your body, are bathed in this extracellular fluid. So we, or your body, needs to maintain a homeostatic environment for this extracellular fluid. And to do that, it has to bring things out. So the extracellular fluid can exchange with the blood, then the blood can go through the exchange cells and move stuff out of the body or move stuff in. Okay, and so that's how we're going to be maintaining this ECF, this extracellular fluid that bathes these cells. Okay, everything inside the ICF is the cellular, or sorry, the fluid within the cells. Okay, so we have ECF made of blood and interstitial or extracellular fluid that surrounds the cells. All right, and then we have the fluid that's inside the cells. So we have this, basically this certain exchange. We have exchange cells that bring, okay, stuff in or out from the extracellular fluid, from the blood and the interstitial fluid that surrounds the cells, and then the cells can exchange with that interstitial fluid, or the ECF. Okay, so we have these series of exchanges. We bring stuff in the blood to the interstitial fluid, and then we can bring it into the cell. The cell needs to get, say, waste components out, brings it out into the interstitial fluid, then to the blood, and then out through the exchange cells. Okay, so we have these series transport across into the different fluids. Okay, but your body maintains these fluids in a homeostatic state within a certain range to make sure that these cells can perform their function. Okay. So let's look again, looking at the fluid compartments. We got the intracellular fluid, that's the fluid within the cells. Okay, and then the ECF, which is made up of the blood plasma, or this purple, this interstitial fluid, okay, that basically bathes these cells. Okay, and we can see that exchange. We have the external environment. External environment doesn't usually, most of the cells of your body, they're not lining the outside and have access to the external environment. So they require that we maintain this ECF, because the ECF is where the cells can exchange. So we need to maintain the blood plasma. We also need to maintain the interstitial fluid in this homeostatic range. Okay, and to do that, we get exchange from the external environment into the ECF, typically to the blood first, then the interstitial fluid, and then from the interstitial fluid we can exchange to the cells, and then vice versa, from the cells to the interstitial fluid to the blood, and then out to the external environment. Okay, so that's kind of the flow of it, and this process here of moving stuff in and out is what's going to help us maintain this homeostatic environment. So here we're talking homeostasis, and you can think of homeostasis, a lot of people think homeostasis, okay, we're staying equal. We're staying equal, right? But it doesn't actually mean equilibrium. Okay? Because if equilibrium, we would have the same amount of, say, salt outside as the same amount of salt inside the cell. So we'd have same amount of salt in the extracellular environment and same amount of salt in the cells. But when you actually look at the chemical composition between the difference between the extracellular and the cells, you can see here's the ICF. ICF, this is what's in the cell. You can see very little the components of salt. Salt is sodium chloride, very little salt, because this is concentration here. Okay, low concentration in the cell, but if you take a look at the extracellular fluid that bathes the cell, you can see there is a high amount of salt, sodium and chloride inside the cell. Okay, and this is actually very important because this is important for the functioning of the cells. Your body, your cells actually maintains this environment where there's high salt outside and low salt inside. And we can look at potassium. Potassium's K. We're going to get into this when we get to the chemistry part, but you can see it's opposite. There's low potassium outside the cell, but inside the cell there is very high amounts of potassium. Okay, so we can see from homeostasis in your body, typically there is a chemical disequilibrium. Okay, there is difference in chemicals from inside to out. It's not equilibrium between inside and outside the cell. We will also find later on, definitely when we're talking about our neurons that you learned about in anatomy, the electrical disequilibrium. The charge, there is more charge outside than there is inside the cell. So there's an electrical disequilibrium between ECF and ICF. Okay. This won't make a lot of sense now. That's osmotic component to most of you, but we will be spending a good amount of time on osmolarity. But there is the total amount of solutes. So the amount of solutes outside and inside, if you took all of what's dissolved in the solutions inside the cell and outside the solutions, and again, if you're not understanding solutions, we're going to be talking about solutions when we get to chemistry, but just basically solutions are a solvent and a solute. Solvent is the liquid, and what is dissolved in it is the solute. Okay, but you can see the total amount of things dissolved in the solution in the cell is the same amount of total things dissolved in the solution outside the cell. Okay, we will be getting, in this case, there is an equilibrium. This is very important that we keep this equilibrium. Okay, but it is not always the case. Homeostasis does not always equal equilibrium. Okay, you see that with the chemical component and the electrical components between the extracellular fluid and the intracellular fluid. Okay, I know these probably aren't making a whole lot of sense, but we will be getting into this when we get into the chemistry. This will make a lot more sense. Okay, but just realize that as of right now, homeostasis does not equal equilibrium. So if we were in actual class setting, I would have you guys group together and come up with some values that you think your body keeps homeostatic. Some we've already talked about, like core body temperature. We'll see ion concentrations in the blood and also inside and outside the cells. Oxygen and carbon dioxide, and that's pretty pertinent with our lungs since that's been in the news these days. Blood pressure, because what do you do when you go to the doctor? They look at your blood pressure. Some others. How about blood sugar? Okay. Urea levels, ion or osmolarity. Okay, these are all things we'll be talking about because most of the semester is going to be involved in how your body's keeping homeostatic, what it's keeping homeostatic. Okay, so these are all common things. When you go in that cold water, your body temperature doesn't drop. It might drop a little bit, but you keep that core body temperature within a range. If not, you get disease, hypothermia. People die of hypothermia, and that's because their core body temperature is getting out of that homeostatic range. Okay, so this is what we'll be spending a lot of the semester is talking about how we keep the different variables in the body in homeostatic range. All right, so let's look how homeostasis is regulated in the body. What process takes place to keep those variables within that homeostatic range. And so to do this, your body uses what's called negative feedback. Okay? And three main components to this pathway: a sensor, integrating center, and an effector. In this case, all right, if a variable is going out of range, right, we have to have the ability to detect this. So we have to have a sensor. We'll use an example is for blood pressure. You have these carotid bodies that respond to stretch. They're called baroreceptors. They're sensors that respond to stretch on the arteries. Okay? And that can tell the body, hey, blood pressure is going up or down. When they get more stretch, it means blood pressure is high. When there's less stretch, it means blood pressure is getting lower. Okay? So these sensors can respond to stretch. You also have sensors that respond to like blood sugar that are in your pancreas. So many times the sensors are associated with the nervous system. Not always the case, but these sensors are there to detect these changes that are taking place in these different variables. So let's use our blood pressure example. So those baroreceptors, let's use high blood pressure. The sensors are getting stretched, and therefore they are detecting a change in blood pressure is going up. The sensors will send that information to an integrating center. Many times that integrating center is the brain or the spinal cord. In our case, there are regions of the brain that are involved in regulating our blood pressure. So those baroreceptors send their information through the nerves to the areas of the brain, the integrating center, that's going to say, interpret it as, hey, blood pressure is going off. So the integrating center can interpret this information from the sensors and send out an instruction. Okay, in this case, we'd send out through the neurons instructions to an effector. In this case, the effector could be what can help regulate our blood pressure. We can change the activity of our heart. If our heart beat more strongly or faster, then we can affect the blood pressure. In this case, blood pressure went off, so the information the brain, the integrating center, would want to send to the heart would be to maybe slow down the pace of the heart and also slow down how strong the heart is pumping. And therefore, if we do that, we're pushing less blood into the vessels and we're decreasing the blood pressure. Okay? Once our blood pressure comes back down within a normal range, okay, within that dynamic range, right? Most of you know it as 120/80. Once we're back within that range, okay, the sensors are going to have less stretch on them, and the sensors are going to send that information to the integrating center. Now the blood pressure is within range. The integrating center does not have to send out, hey, slow down the heart rate. All right, so it negatively feedbacks. We get our effect, and the sensors, basically that effect, can shut down this flow of information. That first, our response was to high blood pressure, and therefore we shut it off. We negatively feed back to it. Okay. So we can see how this would take place in a certain variable. Say, here's our set point for our blood pressure, 120/80, our average. Okay, and remember, homeostasis is not a set point. It's not one flat line. It vacillates within this range. But let's use our blood pressure example. Our blood pressure is going up, starting to get into the high range. All right, and that's where we get the sensors send the information to the brain, and the brain says slow down the heart and beat less forcefully. That brings blood pressure back down. Now blood pressure is going down. Now we're getting within the low range. Okay, now the sensors are, instead of being stretched, they're slacked. Okay, they're slacked, and that'll send the information back up. Okay, we'll go in the opposite range. Once it gets too high, with negative feedback, okay, stops it, blood pressure starts dropping, gets too low, we get our response, and we go back up. Once we hit within the range, right, negatively feedback, stops it, and it drops. So we can see it just vacillates through this variables. Okay. The negative feedback. So it is negative feedback is homeostatic. You might want to write that down. Negative feedback is homeostatic. All right, because once it gets too high, we're going to shut it off, comes back down, and if it starts getting too low, we turn it back on, bring it back up. Okay, so this negative feedback is turning it off, turning it on to keep those variables within the range. All right, so let's use this feedback loop for blood sugar to help highlight this negative feedback and how it maintains homeostasis. Okay, and the yellow dots, we got sensors, purple dots, we got integrating center, and green dot, we got effector. In our good old Western diets, what do we eat a lot of? We eat a lot of refined sugar, so let's just say we're eating some nice Hawaiian rolls with a good amount of sugar, good starch, but lots of carbohydrates, right? Carbohydrates is basically sugar. It gets broken down into sugar, okay, into glucose. So eating, what would that do to your blood glucose? That would increase it. Okay, we're getting an increase in blood glucose, so now we need a sensor to sense this. Okay, the pancreatic islet cells, right? The alpha cells that produce insulin are the sensors for this glucose. They're gonna sense glucose levels are going on. They're also the integrating center because they're going to respond. They're going to send out an instruction, and that instruction is to release insulin. So the effector, pancreatic cells, increase the release of insulin. And most of you probably know, so you've heard of diabetes, insulin is what brings your blood glucose sugar down. It basically allows the liver and allows the skeletal muscles to bring sugar inside. So if insulin's being brought into the muscles, into the liver, we're decreasing the blood or the glucose that is in the bloodstream. Okay, so the pancreatic islet cells integrate, they release insulin. Okay, the liver and skeletal muscles, those are the chief ones that are going to increase the uptake of glucose. And if we're taking glucose into the cells, we're decreasing the blood glucose. Here is the negative feedback. Once we're within a range, all right, is our blood glucose is not going up, it's within that homeostatic range. Well, the pancreatic cells need to increase their insulin release. No, they can shut down that insulin release because now, okay, the blood glucose is within that homeostatic range. Okay, so here we get our effect, and that negatively feedbacks this negative feedback loop. Okay, once we're within range, we don't have to continue doing this to decrease the blood sugars. We can shut it off. And so that is that negative feedback you will see. Okay. On the other end, you forgot to eat your lunch. And if we were in class, I'd say you forgot to eat your lunch before coming to class, but you might be reading this late at night or watching this late at night. So fasting, what would happen to our blood glucose? Our blood glucose would go down. We don't want it to drop too drastically, so we have to increase it. Okay, again, the sensor, the integrating center, pancreatic islet cells. In this case, we don't want insulin to be released. Okay, we don't want the blood glucose to be drawn up into the liver, into the skeletal muscles. We need to increase the release of the opposite of insulin, is glucagon. Okay, glucagon is going to cause secretion of glucose into the blood. So we're going to increase this and less cellular intake because we don't have insulin. And both of these effects will raise blood glucose level. Okay, once our blood glucose level is back within a normal range, we don't have to keep doing this. All right, so that effect, we bring up blood glucose within a normal range and we negatively feed back and turn this pathway down. Okay, so this is how we regulate this homeostasis. This is how we regulate these variables. I'm going to do a couple more slides on this. You can tell this is a key point to the whole thing. I will need you to understand this negative feedback because this is what we'll be talking about throughout the semester. Okay, again, this is just showing you the effects of negative feedback. This is some variable going within the high range. Okay, variable X is going up. Sensor is picking up that change, sending it to the integrating center, and the integrating center is sending the information to an effector that is going to bring variable X down. Once we're within the proper range, we negatively feed back and that shuts down this pathway. All right. In the opposite direction, if we're looking at low range, okay, variable X is going in the low range of homeostatic range. Sensor picks this up, sends that information to the integrating center. Integrating center sends out instructions to an effector that is going to raise variable X. Once variable X is in a homeostatic range, we can negatively feed back and that stops this pathway. Okay, so the negative feedback keeps it from going too high. The negative feedback keeps variable from going too low. It keeps it vacillating within that range. So how does this relate to health? How does homeostasis relate to health and disease? Well, we can get external changes or internal changes that take place. Let's just use organisms in homeostasis. I use my example is you're up at Big Bear and your friends dare you to go out into the snow in your underwear. Okay, there's gonna be an external change. You went from a nice warm environment to a cold environment. Okay, and that's gonna result in an internal change because you at a certain point won't be able to keep up your core body temperature. Your core body temperature is going to start to drop. Okay, organism attempts to compensate. We'll talk a little bit about brown fat later on, but brown fat increases heat. Shivering increases heat, but that's all you got since you're sitting out with no clothes on. Okay, organism attempts to compensate. If you're out there long enough, your core body temperature keeps dropping and dropping. At a certain point, that compensation fails. Okay, the air temperatures, external temperature is too cold for you to be able to produce enough heat to keep your core body temperature up. Compensation fails and you can get illness. In worst case scenario, if you're left out there, you can get disease or death. Okay? But you compensate, you're heating, you're trying to get it, and luckily your friends throw you out a nice down coat. Okay? Your compensation succeeds in that case. You're able to bring your core body temperature back up and you can stay healthy. Keep that core body temperature up. You can forego getting the illness. You might still get a cold or so forth, a disease, but hopefully you won't die in this case. Okay, so that's how we relate. If we're not able to keep within homeostatic range, that puts us in cases of disease state, that pathophysiology. Okay, so we got to be able to compensate for those changes to be able to keep well and keep healthy. So what we've talked about so far is keeping homeostatic. Negative feedback loops keep you homeostatic. Negative feedback loops are homeostatic. I'll repeat that one more time. Negative feedback loops are homeostatic. All right, let's look at the opposite end of the extreme, a positive feedback loop. Do you think negative feedback loops keep you homeostatic? What do you think positive feedback loops do? They're the opposite, so they're actually non-homeostatic. Right? The initial stimulus, in case of blood sugar, what happened? Or sorry, let's do blood pressure. We had a response that brought down blood pressure, and that would bring the stimulus down. Blood pressure would go down, and we would negatively feed back. Here, the initial stimulus causes a response that actually increases the response, keeps feeding back. It's like reverberating. So we're amplifying this initial stimulus. Okay, and most of examples we can use are females have loops that are non-homeostatic for the most part. And those of you that have had a child, you probably have not felt very homeostatic at that point. Okay, childbirth is a positive feedback loop. So let's take a look. We're going to amplify. This initial stimulus actually amplifies, and we get more and more response and more stimulus. So let's see how this works in the case of childbirth. So you can see once it comes time around birth time, okay, this is your classic positive feedback loop because we're going to amplify. Okay, when it comes around birth time, baby drops around lower in the uterus, and we get a stretch on the cervix. Okay, the stretch on the cervix causes release of oxytocin, a hormone. This hormone causes the uterus to contract. That's what we call contractions. Okay, the muscles in the uterus are contracting. That's where you feel the pain. But what do you think that does? It pushes the baby even further down, further against the cervix, so we get more cervical stretch. If we get more stretch, this was the initial stimulus that caused oxytocin. Now we got more stretch, we're going to have increased oxytocin release and even harder contractions. That's going to push the baby even more against the cervix. And so you can see how we keep, and that gives us more cervical stretch and more oxytocin. So we keep amplifying this. We keep amplifying this. Positive feedback loops need an outside source to take away the response. Okay, in this case, what's the outside source is the baby is born. Once the baby's born, that reduces the cervical stretch, and we get the drop in oxytocin release. Okay, so positive feedback loops are non-homeostatic. Another example of a positive feedback loop, again, I'll go in with the females, is when's another time you don't feel very homeostatic? Okay. Maybe during your menstrual cycle, because that is a positive feedback loop. Ovulation is a positive feedback loop. And most the PMS symptoms, those are non-homeostatic, and therefore it is a positive feedback loop. Once the egg is released, that loop is shut down. Okay, but these are non-homeostatic. Negative feedback loop, homeostatic. Positive feedback loop is non-homeostatic. It amplifies the signal instead of turning the signal on or off to keep it within a certain range. Or not signal, stimulus, sorry. So what are modes in maintaining homeostasis? There's regulation. Okay, this can occur, we can have intrinsic or extrinsic regulation. Okay, in intrinsic, okay, cells of the organ sense the change in signaling neighboring cells to respond appropriately. Okay, this is intrinsic to the cells. The cells are regulating their own response and the neighboring response. So this is very local control. Many times your arterioles that regulate blood flow to certain organs, your organs will regulate the blood vessels that are near it, but only locally. It won't regulate the blood vessels that are far off in a different organ. It will just regulate the blood vessels that are neighboring it. So intrinsic control is very local, okay, response, and it's controlled by the cells around in that area. Extrinsically, it can happen over longer distances, and typically it's the brain that's going to regulate other organs, or endocrine system, like the cells of the pancreas regulate the cells throughout the body, not within, right around the pancreas. It regulates cells that are far off in distant parts of the body. So extrinsically occurs over longer distances. Control can happen from the brain all the way down. The brain can control factors that are taking place in the toe. Okay, so this typically for extrinsic control, we're going to utilize the endocrine or nervous system to control the activities of the cells of organs throughout the body. So the next slide here, it's kind of a rehash, or should be a review from anatomy. So I'm not going to spend a ton of time on here because this is stuff you should have seen and should have in your bank of knowledge here. But let's look at levels of organization of an organism. Okay. And going from smallest to largest, we can look at atoms. Okay, atoms such as carbon, such as sodium, chloride. We've seen those two already. Those are atoms. Atoms are put together to form molecules. Okay, multiple molecules put together form cells. So this is the basic functioning unit of the body. We're going to spend a lot of time on cells. Multiple cells put together to form a functioning tissues. Now you did all that histology and anatomy. You were looking at the different tissues with the different cells in them. Tissues, multiple tissues put together to form an organ, and then multiple organs work together to form an organ system, and then organ systems are put together to form the whole organism. In anatomy, you spent a lot of time up in this range. Okay. Where do you think in physiology we're going to be spending? Again, we got to use chemistry, because chemistry is what makes the body go around, what makes things work. So we're going to be spending most of our time with physiology. We're going to be down in this level of organization most of the time. Okay, we're going to be looking at the different cells of the different organs, but to see how they operate, we have to they operate by using chemical reactions, which involves atoms that turn molecules. Okay, so we'll be spending most of our time down here, and I know that word chemistry, it's a scary word for some of you guys, but bear with me, we'll get through it. We're going to spend a whole chapter on chemistry and looking at chemical reactions. And again, what I've told you guys before, if you're not understanding something, send me an email. We can talk about it. We can go in, I can do private groups to go over stuff, and I can't always see whether you guys are grasping this stuff, so you got to make sure you get a hold of me to make sure. So just a prelude, we're going to be spending most of our time in here because we're going to be dealing with chemistry. Okay. Let's look at the tissue levels. Hopefully you all remember the 4 different tissues, but crazy thing is we have 36 trillion cells in our body, and they're all basically made up into 4 different types of tissue. We got epithelial tissue, that's going to form the coverings and our linings. Okay, these are the ones that are exposed to the outside world. Your skin, your digestive tract. These are all lined with epithelial, and epithelial lines as faces. So this is basically our preface to the external world. We have connective tissue, kind of the glue and filler of the body, muscle tissue, and then nervous tissue. Okay, probably the most we're going to be spending a lot of time with epithelial, with nervous, and muscle tissue. We won't be spending so much of time with connective tissue, but it is an important. But since we got to regulate things coming in and out, we got to deal with epithelial. We're going to spend some time on muscle, but we're spending a lot of time throughout the semester because we have to keep things regulated. We have to have a system. We have to have an organ system that allows control, to allow integration, and that's going to be your nervous tissue. So throughout the semester, we're going to be dealing with nervous tissue. Okay, and we'll be seeing lots of epithelial, but that's where we'll be focusing. But again, we're going to be focusing more at the cellular level and molecular level when we're dealing with physiology. Okay, just a refresher on epithelial tissues. Those are linings and covers. Also makes up the glands. Okay, the glands are the endocrine glands are also going to be another modes of control. The nervous system and endocrine. So again, we'll be talking a lot on epithelial tissue. Okay. What are some functions? Well, it allows us physical protection like our skin, but it is the face to the outside world. It is the tissue that basically is exposed to the outside world. Like your heart is not exposed to the outside world, but epithelial tissue that cover the linings, they face the outside world. So diffusion and filtration, this allows to get things into the body or out of the body. Secretion, things out. Absorption, sensation, and ion transport. So these epithelial tissue have an important function. These tissues of the cells of the epithelial tissue serve an important function. So we'll be talking about them pretty extensively. Okay. Like I said, the connective tissue, we won't be spending a ton of time. It is an important structures. It makes up important structures, but we won't be spending a ton of time talking about the different connective tissues. All right, we'll be focused a lot on epithelial, a lot on nervous, and some on muscle, but very little on connective tissue. But again, all this tissue is important. Muscle tissue we'll spend a fair amount of time on. We have a whole chapter going over muscle tissue. All right, and if you remember, there's 3 different types. The one you're probably most familiar with is that skeletal muscle tissue, okay, that control our limbs, give us movement, so forth. Another important one, the cardiac muscle. So we'll be spending a lot, the chapters on cardiac, so we will be talking specifically about this muscle. And then smooth muscle. Okay. We'll spend some time on this too. Okay, we'll have a whole chapter talking chiefly on skeletal. We'll spend a lot of time with cardiac when we're going to the muscles, and smooth muscle we'll find throughout because it's lining the gut and blood vessels. So we'll also see it in cardiac also. Right, neural tissue. This, and I come from a neuro background, so this is an important one to me, but it's important throughout the body because most of the control, what's helping control the homeostasis or controlling the organs that are going to allow us to maintain homeostasis is controlled by either neural tissue or endocrine, but fast control is going to be done by our neural tissue. So hopefully you guys remember your neuron here. Okay, and basically in that spinal cord and brain, and then we have all those spinal nerves that attach or are able to go out to the different organs to help send signals to them. Okay, so we will spend a good amount of time on neurons, and then we will also see neurons throughout because they are talking to all the different organ systems throughout the body. Okay. And we also, besides the neurons, we have the glial cells, so we'll talk a little bit about them, but we'll be a lot of focus on the neurons, the ones that actually carry the information and signals to and from the nervous system. Okay, if we put it all together, we went from atoms, from those carbons and oxygens and so forth, put those together to make a molecule, put multiple molecules together to make a cell, and then multiple cells to make tissue. And then we put multiple tissues together to form an organ. In this case, we're using the skin. We got epithelial tissue. Okay. Connective tissue with our dermis and hypodermis. We got muscle, smooth muscle with our erector pili, and then nervous system to give us. So we've got all 4 tissues here to make up this organ, in this case, which is the skin. Okay. And then ultimately, we put multiple organs together to make an organ system. Okay, our digestive tract, we got multiple organs here serving different functions to serve the function of digestion, which we will cover pretty late in the semester. And then multiple organ systems put together to make the full organism. Okay, but again, if we were in class, I would have had you guys give a refresher on which the different organ systems. And so we're going to spend, many of these systems are here to help maintain homeostasis. Okay, so we are going to spend a good amount of time. The only one we probably will not be touching on most is integumentary, and then reproductive. If we have enough time, we're going to be hitting reproductive a little bit, but all these other systems, these are all involved in regulating homeostasis. So we will spend a good amount of time on these different systems. But again, we're not going to be talking at the system level. We're going to be talking at that cellular level, molecular level. So again, I keep stressing it, but we gotta get the chemistry down. The other one I really want you to get down is the membrane transport. Okay, I'm going to harp on these because these are things you have to learn, because if you understand the basic concepts, you're going to understand them in the concepts of these different organ systems. Okay, so we'll go through these different organ systems and looking at how the cells in there function and help maintain that homeostasis. Okay. Just a summary of them. So this should all be reviewed. I expect you guys to know a pretty fair amount of these. Okay.